Good day, everyone. My name is Soremekun, and I'm presenting a research titled Genome-wide Association and Mendelian Randomization Analysis reveals the causal relationship between white blood cell subtypes and asthma in African population. White blood cell traits and their subtypes like lymphocyte count, monocyte count, eosinophil count have been shown to be associated with quite an array of diseases like cardiovascular diseases and stroke. So the aim of this research was for us to identify the genetic drivers of white blood cell traits in African population and subsequently use these genetic drivers as instrumental variable in a Mendelian randomization analysis to determine the causal relationship between white blood cell traits and asthma in African population. For us to do this, we used two independent African cohorts, the APCDR and BCX, which we meta-analyzed and did a Bayesian fine mapping, and we further went ahead to do an Evans variance weighted Mendelian randomization. For us to do this, we used the lead SNPs in each of the white blood cell subtypes as genetic proxy for the exposure. While for the outcome, asthma in this case, we used individuals diagnosed with asthma in the African American Kappa cohort. Figure 2 shows the Manhattan plot of each of the white blood cell traits, showing the significant SNPs at the p-value threshold of 5 × 10⁻⁻⁹. While table 1 shows the Mendelian randomization analysis result. Of note is the p-value column, which shows that monocyte count, neutrophil count, and white blood cell count are all statistically associated with asthma, unlike lymphocyte count that shows no statistical association. And another interesting thing about Mendelian randomization is for us to determine the direction of effect of our phenotype. In this case, monocyte count, neutrophil count, and white blood cell count all showed a negative causal estimate, which suggests that rather than being protective, that these traits are rather causal for asthma, unlike lymphocyte count that has a positive estimate which shows protective effect against asthma. In this research, we've been able to identify 5 genes that are identified for the first time to be associated with white blood cell traits. And again, we saw strong causal relationship between monocyte, neutrophil, white blood cell count against asthma. And this result is quite consistent with the result coming from the European population. I would like to appreciate the member of the African Computational Genomics Group. I would also like to appreciate MRC, UVR, London School of Earth and Tropical Medicine, without forgetting CHPC for computational resources. Thank you for listening.